every electronics was made in Malaysia. So Japanese companies had set up the, their factories in Malaysia. Even today in Penang, that's another island. Lots of semiconductor industry has their fabrication facilities. All right, so just give me one moment and we'll get going. So we'll continue our discussion where we left off on basic biological molecules. I want to get into a little bit on DNA. DNA, and the reason is because that has the genetic code. How do we handle DNA? What is the significance of DNA when we talk about nanotechnology? That's some interesting questions because a lot of work happened and discussion interface of basic biological molecules. I want to all right. So nucleus is the largest body in a cell, and it has chloroplast and mitochondria, not inside. So it's like them, and the difference then the other things is that this contains the genetic material and protein synthesis machinery in it, and but they're not independent from other things that are in the cell, but a lot of proteins are encoded by nuclear DNA, and they're made in cytoplasm, and then they go wherever they're supposed to go. So nucleus is the largest and most important organ or body inside the cell, and the most important thing, it contains the genetic material called DNA. Right now, things go in and out of nucleus also through very small holes, pores, nuclear pores. So these are channels, and the regulation around or through those channels is again a part of this membranes that keep it together. Now, why we are interested? Because a lot of work started in nanobio interface was on the detection of specific genes. There's history of genes with a very large scientific project that happened all over the world. It was called Human Genome Project. We'll talk about that also, because that project helped us not just learn so many genes, it helped us sequence the whole human genome. Like we could find out exact sequences of human genome. Now, nucleic acid is a class of molecules. If it is, so there are two types, DNA and RNA, deoxyribonucleic acid and ribonucleic acid. So DNA is what we talk about that contains the genes, but RNA is a complementary thing. What it does is it has its own role in the whole process. But we have to look at what is the basic makeup of a nucleotide. So when we talk about DNA, we are talking about many, many bases that come together and form a long chain, create a long macromolecule. And each unit that makes up that long DNA molecule, we call that as a nucleotide, right? So the basic unit in DNA is called a nucleotide, and the nucleotide has three basic parts: a sugar ring, a phosphate group, and a base. So these are again chemical structures. So we identify one structure and we call it a pentose sugar, right? Because it is sugar ring. The base itself are important because there are four fundamental bases. There are four fundamental configurations of those molecules that we call bases. The names are adenine, guanine, cytosine, and thymine. We just call them AGCT. When you deal with DNA, we'll be dealing with four bases. A will always make a connection with T G will always make a connection with C. So they are complementary to each other. So there's sugar, phosphate, and base. When they come together, we call them one nucleotide. A, C, G, T, whatever that uh, base is, it would always carry with it sugar and phosphate. And now these bases, it's an interesting molecule. The bases bind with each other through hydrogen bonding, but they're the what you call the sugar and phosphate, they would be binding with each other in the longer way along the length. But two single strand DNA molecule will come together, and A will bind with T and G will bind with C. This is something I cannot emphasize how amazing this is, that this selectivity is at the very fundamental core, that G will always bind with C and T will always bind with A, which means that if I pick one side of DNA, I can always know what's the other side, right? So where I have an A on my one side, I can say there is a T on the other side. If I have a G, I would know there is a C on the other side. That's the normal what do you call configuration of them. And these two strands, you can separate them out, so that would be a single strand DNA. And when you get a perfect complementary, you bring them together in solution, they will find each other and they will create this double strand duplex that we see in movies or pictures, and I'm showing you here. So at the heart of it, at the bottom of it, is this amazing selectivity of G always binding with C and A always binding with T. You can say it the other way around as well, right? Now, that's a basic configuration of a nucleotide. And then when they come together, they create. Now, when we talk about RNA, we just have a very small difference, that instead of T, we have U there. That's all it is. That's the basic difference. And when we write the sequence, we would replace that. Instead of writing T, we will write a U there. Can you please start slideshow? Hold on. Sorry. So I think I'm sharing the wrong screen with you guys. Is this okay? Is this the slideshow, right?
Yes, sir.
Yeah, I'm sorry about that. So yeah, thank you for bringing it to my attention. You don't have to write in chat and wait for me to look at the chat. You can just speak up. I'm sorry about that. Yeah, I should have been more careful. All right. So now this double strand, there's some basic features. As engineers, what excites me, because I can use those features to design a sensor. I can design a device to look for cancer genes. I can look for, in an unknown sample, the race is that how can we make devices and systems that can cheaply and quickly find out the whole genetic makeup of a person, right? It has strong value, because if I get my genetic test done, and if I find I have cancer genes, I don't have cancer, but if I have cancer gene which are running in the family, I can change my lifestyle, right? So that's the goal, or that's something which has been driving a big industry for last 20, 25 years. Now, so it helps us learn a little bit about that. There are physical parameters in these biomolecules which can design in our devices, right? So there is a new area called biophysics, which is nothing but how do we quantify mechanisms in living things, right? So hence some physical features. So it's a double strand helix. Now, when you talk about this, the sugar ring we were talking about, so there is a nomenclature of which we give numbers, specific numbers to those carbons. carbon number 1, number 2, number 3, number 4, number 5. Now, every, what do you call, the base, one base has 3 and 5 of carbons which bind to the next one. So 5 carbon would make a connection with 5 carbon of one molecule. One monomer will make connection with 3 carbon of the next one. So if you look at the long chain, there is 3 prime side and 5 prime side. So we call it because of this sugar backbone which runs over the whole length. So this is a feature of the molecule that we say 3 to 5 or 5 to 3 sides. So these molecules are two strands. They are exactly the same, but when they come together, one side has to, we call them anti-parallel. So one is if going 3 to 5, the other one is going 5 to 3. Right? So if you see the word anti-parallel, the idea is that their makeup is something we run them in the opposite direction. They are like that. The distance between two bases not from the opposite base, but along the length, that's 3.4 angstrom, which is 0.34 nanometer. So if I'm making a device where I have electrodes which are say 10 nanometers apart, I would know exactly what length of DNA I can detect. If there is a DNA length smaller than that, and I'm thinking to bridge them, we can exactly calculate what would be the physical length of a molecule given if we know how many bases are there, right? So, and the diameter of DNA is 2 to 2.2 nanometers. So we take it 2 nanometer. So that also helps us as device designers if we are thinking to use DNA in something, right? So just a bit more. Now, maybe some of you read on discoverers of DNA looking at KPR. So this is 1953, they published a paper. And if you read that paper by Francis Crick and Watson, it's a page and a half, very small paper. They got data from 2 other scientists, Franklin, and there was another guy because they got 3 of them. So Rosalind Franklin was working with Maurice Wilkins, another person, and they got the data. They did something what you call X-ray diffraction. That data they shared with them, and Crick and Watson were theoretical people. So they came up with the theory of what would be the angle of bases and how would this molecule be in nature, looking at that X-ray diffraction data. So on that, then the 3 of them shared Nobel Prize in '62. So about 9 years later. Now, but this lady, she passed away before 63, but she's known to be and recognized to be the actual person who got the data from those DNA molecules. So think about it, that X-ray diffraction technology is so powerful, and it's old technology where you can see a single molecule or get an image of diffraction image from a molecule. So what happens is they put a sample and we throw X-rays on those DNA fibers. So you have to find the right spot. And all the DNA, we try to put them in all the same orientation. Now, when the X-rays, they strike these bases, they get diffracted. And we image the diffraction pattern that comes because of those diffraction, because of the striking with those bases. And that pattern of DNA is what was then used to come up with the theoretical model of how they should be. Now, why is it important for us? Because this is the physical and functional unit of heredity. This is how the traits transfer from generation to generation, from parents to offspring, in all living things, right? Now, if you look at the whole DNA, it's like in human DNA is 3 billion bases, but not all parts code, or not at least that we know, they all code information. Most of it, we don't know. We call them exon and intron. So basically the part that we believe that has nothing coded in it, we call them introns. And the parts that we know they code for proteins and amino acids, which give properties to cells, those are called exons. Now, I have taken next step in this. From DNA, yeah, DNA is just a storage of information. From DNA, we create proteins and amino acids. So those proteins and amino acids are who do functions and give properties to cells, right? So that's a step what we call translation, going from the storage to actually making those proteins that give physical properties. That's a step which is called translation. And what we do is three bases come together. We call them triple codons. Three bases come together, and they would result into the machinery, whichever will take that DNA and make a protein out of it. Those three will tell that machinery what amino acid need to come. Amino acid is another macromolecule. Those amino acids would come together based on this thing is reading up DNA, that small gene, whichever has come out, and that results into the formation of amino acids that come together in specific format or specific sequence. And that's a protein. And that would fold on itself or do and go on and do whatever it's supposed to do. Now, these amino acids, when they come together, they form a protein. They form all the living things or all the properties that are there, right? So that's another part of interest for nanotechnology, that can we identify that process, or if there is a break or something not right in that process of translation, can that be detected? For bad cells, for cancer cells, can we disrupt that translation process so it doesn't replicate, it doesn't grow, it doesn't eat away the mutation? So it's a whole bunch of nanotechnology or interest of nanotechnology to go and do something about it in translation part, right? So a little bit about Human Genome Project. So we knew bases, we knew, okay, we knew some genes, but then it was an international push where people from all over the world came together. And they, and today we have identified 30,000 plus genes, right? And many of those have been direct result of this work. And the goals were, if you, I put the italicized, I have italics, put the words in italic, that all genes of human DNA, the data was collected all over the world in so different labs. And they made a, and we're talking about 20 years ago, right? More than 20 years ago, it completed about 20 years ago. It started in 1990. In 13 years, they finished. They started sharing data in real time. So one lab would sequence some part of human genome in Japan. They would upload the data that became available to everybody in Europe, in USA, and whatever country were involved in that. That had a major push in the computation tools also we had, what we have today. Because there was a race of how we get the data, we put it in the computer, you want to find the patterns, right? So genes are patterns, which we find in, for example, if you find some pattern in 1,000 people and they all have gray eyes, probably, just give an example. So we call them alleles. So those subpopulations which would share those sequences and share physical properties, so then they would be identified as genes for that particular kind of traits or physical properties, right? We'll go over a video at the end on actually how it was done. So I put the link here. We'll go over that later on. But before we do that, let's talk a little bit about the DNA process of DNA replication. Cell divides, right? So during the cell division, the DNA is replicated, two identical copies are made. And then those two copies become part of those new baby cells and they go on. We do this in the labs as well. So we do DNA replication in lab. You know, when they show in the movies that somebody took a DNA sample is taken and then they sent to the lab. So the basic idea is we get small amount of molecules from somewhere. It's not enough to do processing. So we make copies of that. You know, like you take one page and you can make 1,000 photocopies of that, which would be exactly the same. It's a similar idea. We get few copies of sequence somewhere. We do the same process as it is done in cells, very much the same. Now we do it in test tube and we make copies of DNA. Now this process is very precise. And it happens because there is lots of parts which are extra, right? So there is the chances of error become low because many of those parts don't contain any genetic information. How it happens is there are the two strands which are running, they are separated. Now, when they are separated, you can pick any of those. You can pick one or the other because they are exact complements of each other, right? You have to be careful. They're not copy of each other, they're complement of each other. If I know one, I know automatically the other one because A goes with T, G goes with C, right? Now, when we do this, we unwind and those two sequences now are our templates. Our templates, now we can use those individual ones to start building the other's chain with each. So hence what we do is we keep on adding nucleotides. So by taking one parent, like the blue one, I can split them and I can make a copy. Again, copy here is the complementary of that. And hence I can get two exact copies of the parent ones from there, right? So those bases, we got the bases, we have the bases, and then we have other molecules and enzymes who can read G and they can put a C there. They move on. Who can read whatever base is there and they can, from free floating solution, they can get the next sequence and put it next to it. So it's a very exact science how we take one DNA molecule and we replicate. And it replicates in both directions. This parent sequence or even a small sequence, if we are amplifying a smaller sequence, we take that smaller sequence and we use that strand as template, and then we use, so that template or the replication would then be something which would be used to make copies of the original one. And then we have a molecule, an RNA molecule, which we bind as the starting point, primer. And then this enzyme comes and that starts building on top of that primer, and whole new DNA is synthesized by this molecule called DNA polymerase, right? So it builds from 5 to 3 sides. So we add to the 3 end of the chain. And at the end, we can bring the fragments back together by another molecule called DNA like this, right? So I don't want to scare you with these big words. I just want to give you the big picture that we can take the parent molecule and make many more copies with that. And it's a very, very well-defined kind of a reaction, and we engineer that reaction with that. It is called polymerase chain reaction, PCR, where we would take few copies and we can make multiple copies of that DNA. We keep on doing the process until we have as many copies as we want, right? So now this, the polymerase, DNA polymerase, the most important component, the molecule that goes in it was identified by Dr. Mollis, who got Nobel Prize for that. So it's interesting how this DNA polymerase can is a building machinery that you give it the template, you give it the primer, and then it starts taking free floating bases and start putting the exact base that's supposed to go there and makes up the whole DNA, makes up a copy of the whole DNA right there. Now in lab, we do this with this process called PCR. 3 steps. We take the original double strand molecule. Now there's another feature to it that these molecules, they are bound together because of bases, right? And bases have hydrogen bonding between them. We know specific, we can calculate exactly what is the free energy associated with that. And that tells us that how much energy do we need to give to break those hydrogen bonds across the whole 2 chains. And that tells us melting temperatures. Exactly like we use the word for metals, that we have to heat them above certain temperature, after which they would melt, right? They would change phase. Here, what we mean is we know exactly what temperature do we have to heat that solution of DNA where double strands will break apart and become 2 single strands. So that's called denaturing or melting. Once you have those 2 single strands, we add the primer, we put the 4 nucleotides free floating, bunch of them, and we add DNA polymerase that starts the production of new strands. All right. So kind of an engineering kind of step where we have denaturing and adding the primer, which would bind at some place, and from that some place it would start building DNA molecule. Right. So when we do in this PCR reaction that we are talking about, so that's where denatured. The second part is called elongation, where we are now making base space, and then we do annealing, where we want to bring the free floating, all the new copies of DNA, we want to bring them together and create all them into double strands. So just a picture of, again, we start with 2 blue, split them, put the primer, add all free floating bases, bring in the DNA polymerase, it starts building from that blue, it starts building the red ones. So from 1 double strand, we got 2 copies made, from 2 to 4, 4 to 8, and go on, and so on. So it's a very straightforward process. Now I want to go into why are we talking about it? Why are we studying about it? Because we have done lots of work in miniaturizing these processes. This is an example of PCR on chip. So PCR machine is a huge, big machine on the table. But we can do the same thing now on a chip, right? It's an old example. How old is 2004? What we are doing is this concept of lab on chip. It says PCR on chip. PCR is because this chip is doing PCR. But there's a general term, what we are getting into is called lab on chips. The idea is that whatever you would do in a big lab to do medical testing, reactions, we can do on a chip. The chip can be made to work with small amount of input materials. We would use very smaller amounts of reactants, whatever things have to go, and we would get results very quick, right? So in this case, PCR, like I was talking, 3 steps: denaturing, annealing, elongation, denaturing. We can do all those things on the chip where we can have mixers, MEMS. We have studied MEMS. We can have valves where we can push fluids around from one part to other part. We can pump things. We can run them through channels. We can make all kind of things at micro scale to deal with nanoscale biomaterials, right? Now, this is just an extension of, we talked about PCR and wanted to show how is that. We can interrupt the whole process of translation, right? So I was saying, okay, we have DNA nucleus. It comes out and we it comes out, the DNA splits apart, and just like we do PCR, so inside a cell, molecules, they come together and they make a copy. Again, copy is the complementary. They make a copy using, so it's an RNA. So instead of one of the bases, there is a different base, but it is exact complementary copy of that. That mRNA comes out of nucleus, and then machinery comes together and reads that mRNA, makes tRNA, but from that RNA, it creates proteins, which can stay inside the cell, get to the cell wall, go outside of the cell. Maybe it's a signal you want to send to other cells. So all these steps we can interfere, we can detect, we can identify, we can read. And that's where the potential, and that's where the big work has come through from the nano side. And over years, there's more and more things are happening where we want to maybe detect mutation. We all know mutation is what we know, it causes cancer. We want to detect protein misfolding. That's now known to be one of the reasons for Alzheimer's disease, right? So we want to detect even a single base mutation, single base sequence variation, right? They are called SNPs, SNP, single nucleotide polymorphism or something. We want to identify and look for maybe an unknown sample. Where is it coming from? Is it, which species does that belong, right? So these are all real world challenges which have been addressed and are being done more and more with nanotechnology, with chip-based things, right? Now, on talking about the PCR chip, now, yes, there is some processing that needs to be done away from the chip, right? So you have to purify your samples or whatever, the biopurification, and then we have to inject that. So there is some pre-work or there is some post-work also, because you have to now read it and you have to make an interface with computer. Okay, but the idea is that the whole need for a clean lab or a chemistry lab is not there anymore, which now means you can deploy this in remote areas where you don't have trained technicians, maybe. You can use it in places where we don't have infrastructure, we don't have big hospitals, but if you can make a lab of a chip, kind of a standalone system, just like computers are everywhere now, right? They may not have clean water, but they have computer and they have cell phones, they have connections, right? I all over the world, there is more connectivity. There's more feasibility to deploy these kind of systems, even when there are resources less or there are rough places and whatnot, right? Now, going into a very interesting direction.
Sir?
Yes, sir.
Sir, what is the meaning of DNA sequencer, sir?
DNA sequencer. So DNA, there are 3 billion bases, right? We don't know what is the sequence. We want to find out what are the sequence, because if we find the sequence, those 3 billion, think about if we have 3 billion alphabets. I don't know what's a good example. So suppose we have this die, right? So Ludo, we play Ludo, die has 6 faces, right? Suppose we have something with 4 faces. And now that can be made, if you make one edge triangular, you can have 4 faces kind of die. There are 3 billion of those, and they have some order, A, G, C, T, T, G, C, A, whatever. But we don't know. Why do we want to know? We want to sequence that. Sequence is we want to find out exact sequence. That's called DNA sequencing. And a machine or a system that would do is called DNA sequencer. Why you want to do that? You want to find out genes. Those genes tell us today hereditary, where we come from. People find out they are from this descent, they are from Turkish descent, like we are Mughal descent, right? Mughals are supposed to come from Central Asian Republic, Turks. Turks also came from the same area. Aryans. So these are races in the world, African races. So we can find that. We can find genes related to diseases, right? We can find out for law enforcement if there is some biological sample at a crime scene. We can take the culprit, we can take the people who we may think are the culprits and we can match the sequence. So we need to get the DNA, sequence it, and then compare it with the sample that's found, right? You get some
Sir,
idea?
can only the Oxford nanopore is the only device to detect the DNA sequencer, sir, or is there any
Oxford
other?
Nanopore is one of the companies who is trying to do it with in a different way, but there are very well established systems who do sequencing.
Same at nanoscale level, sir.
Nanoscale what?
Same at nanoscale level, sir.
Nano,
I
okay, so there is a well-established biochemistry process for DNA sequencing, right? Now, and Oxford Nanopore is one of the companies. There are so many other companies who are trying to do it where they want to interface at single base level or a few base level. But think about a base to base distance is 0.34 nanometer. How can you make something so small unless it's at atomic scale? But then you have to interface with the outside. So there are issues, there are opportunities, but there are many companies who are trying to do it. If you throw one company or one technology to me, I'll probably don't know. I'll have to look up how they are doing it, right? But there are so many companies, so many places who are trying to do that quicker, faster, right? And of course, that's where nanotechnology comes in, where we want to interface or we want to read those things which are so closely spaced to each other, right? Going from one base A to C is 0.34 nanometer, 3.4 angstrom, very small. Or maybe data analytics, maybe you want to read 2, 3 bases at a time and then uh, do some signal analysis and figure out what were the sequence, what are the bases in that small part. So there are many ways how they're trying to do. Right? Now, what happened about 20 years ago is the idea of DNA microarrays. DNA microarrays, the idea is that, okay, we know the sequences. We have a database of genes. Now, if I get an unknown sample, I want to find out if there are at least those known genes, are they in that given sample or not? So from known genes, what it says probe molecule here, right? So you see those blue spots on the chip, they are probe molecule. Those are known sequences that we already know that this sequence would be a gene related to, I don't know, lung cancer, for example. There are mutations, known mutations. There are lung cancer, breast cancer, there are prostate cancer, the pancreatic. So there are many, many known mutations for all those things. So we know genes related to those illnesses. So we know we put the molecule there, probe molecule, known molecule. But now this unknown sample that's coming from this test tube, we run over this chip. If those sequences were there that we are looking for, they will bind to the blue spots, right? If they will not bind to blue spots. Now the binding of those molecules is what we can detect. So there are dye molecules, dyes which give color. So there are dye molecules which give light, and they detect. For example, one of the ways they detect double strand formation. So you had single strand put on the surface, like in this one, and then you brought the other one, it bound, and you can measure the binding. At that binding, you can look as a photon coming out of it. So we can see, theoretically, we can see single molecule binding. If that happens, this means that the given sample, and that sample came from a person, for example, has unknown gene for, I don't know, for cancer, for example, right? So it makes sense. So this became a really powerful technology, which is now gone into do so many other things now. So you got the idea of base pairing that, yeah, A was supposed to go in T, G is supposed to go to C. So we know the known sequence, we put them on the chip surface in the blue spot, right? So there are all these thousands of single strands are now, they're waiting for the sample to come in. We put the sample in, we let it settle down, bind it, do some temperature cycling or whatever. If we wash off what is not bound, it will look if there is any signal, which would mean that those known genes were there in the system. So DNA microarray is not sequencing. It is just looking for some very specific sequences to look for if they are there or not, right? Right, questions?
Sir,
Yes, sir.
sir, can we able to store the result or copies of DNA, sir, for long terms like year or 2 years or more than that, sir?
Say again. So you're asking if we can store the DNA sample itself, the molecule.
Yeah, yes, sir.
You put it at minus 80 degrees centigrade, you can store it forever.
It means it needs some
Forever, but
temperature.
it's known 5, 10 years, 20 years. Yeah. Yeah. So you have to freeze it because there are molecules in nature who would digest and eat it. So we want to clean it really well, get rid of all impurities, freeze it. You can, yeah, theoretically, you can freeze it forever. Right? Yeah. Now, but what's the interesting part here? When you run DNA microarray, you know which sequence is there. So we can look up the database and you can find the sequence. The sample is irrelevant. Now we have the data now for about that sample, right? So that data is what becomes important. And that data is nothing but AGCT coming in specific orders, a specific pattern for those 4 bases, right? I cannot emphasize enough how amazing is this in nature by the creator, that 4 bases can encode everything that I'm doing, looking, or you or each one of you in this room, how you're different because of those 4 bases coming in different sequences, right? So again, another thing, we don't just look at the DNA that's there. We look at RNA also. Now, if you look at RNA or messenger RNA, we are looking at expression, another new word. So DNA is stored in nucleus, but not all genes get to be expressed. The word is expression. That's a technical word, which means that, oh, I have 20 currencies in my table, right? From, I don't know, Turkey, Poland, Dutch mark or Dutch, they don't make Dutch mark anymore, but from Switzerland, Malaysia, whatever. I don't use all of it, right? They're stored, they're saved. I may use dollars, I may use rupees, right? I may use Bitcoins now. So, but just a very crude example. So those genes would be there. It would be more interesting if we see which genes are getting expressed, because now we would know what actually cell is going through, what kind of proteins are being made, or are there any proteins or any mRNA being made that will result into abnormal function of the cell. So we can take mRNA also, but mRNA is again copy of the DNA which was stored. But now why mRNA is important? Because mRNA, if the cell is making mRNA, it means those genes are getting expressed. We take mRNA out, unknown mRNA, we don't know what's in there, but we know what would be the sequence of the bad genes. But mRNA itself is kind of, it's not stable. So we make a DNA copy. Again, copy mean complementary of that. And we use that DNA to now look for. So again, we do this by the same process of using a primer and DNA polymerase to create a DNA chain. So here it is called cDNA, right? Complementary DNA. So the idea is now we want to look what is actually being expressed. And then we run it on DNA microarray and we get the expression of whatever was in there, right? Couple of more things, and then we can stop. There are very interesting special properties of DNA. I apologize for this dirty looking slide, but what I want to show, and I'll take you through one by one. Some things we have already discussed. So one sequence goes, if 5 to 3, the other one goes 3 to 5, right? Again, they are the base, the sugar ring carbon placement, 1, 2, 3, 4, 5 carbon, 3 carbon, 5 carbon, they go on the other side. A is going with T, G is going with C, so I just wrote it with my hand. When we apply heat, they melt. So each sequence would have its own melting temperature, depending on its sequence and depending on how much salt we are using. So DNA molecule is suspended in aqueous solution because we have to give it salts. We have to give it positive charge. Why? Because phosphate groups are negatively charged. Two strands would not come together. They will repel each other. So we have to dip, we have to have aqueous solution, for example, KCl as written here, to have positive charge available to shield this negative charge of the backbone. So that's called shielding charge. When we do that, when we have those, then two molecules can come together and form double helix, which means the nucleus, if it is containing those double strands together, it has enough positive charges there to shield the phosphate backbones from each other from two strands. Hence, they come together. And when if you have to break them, all the system has to do is remove some of these charges, and negative repulsion will result into breaking of double strand from that particular part, wherever the gene is supposed to be translated, right? So that's the physics side of it. Now, we can create many different shapes and structures using by engineering specific bases. For example, here I've written, I call it hairpin loop. It's how what we call it. So the idea is that I make a sequence such a way that the edges or the end of that sequence, I make them perfect complementary. If I put them in solution, they would find each other, those edges, and they will form something like that because those far away parts are complementary. So we get, like ladies put the pin in the hair, the end of the pin is loop, small loop. After the loop, it comes together. That's exactly what we call it, hairpin loop. These are used for signaling a lot of time, right? And it says hairpin loop. If it opens up, if you give it enough heat, it would become linear DNA again. So there are few physical things that we play with DNA molecules to make reporter molecules. So for example, and we can, when you do that, okay, you can order DNA of whatever. So they make synthetic DNA. Same basis, but built in a lab. So if you know a specific sequence we want, we can order it. There are so many companies and they're very cheap. There are machines called DNA synthesizers who do. I can get a sequence with just AAAs. These are called poly sequences. So because you have one thing which is covered, the whole sequence is one type of base. You can get poly DC, poly DS. So D is just saying that there are many of them. You can order palindromes, right? Just like we write English, you can order something which runs the same way from both directions, which would mean you don't need, so each molecule is complementary to itself. You heat it, they will all come together. You don't need a complementary sequence. So there are lots of interesting plays we can do as engineers to make our DNA molecules for whatever application or experiments we are doing. All right, so it's another interesting application. We have lots of functional groups that we can use. We can add those functional groups two DNA molecules. We can put them at the edges. We can put them in the middle. So here there's an example. It's called a molecular beacon. There is a special name. The idea is that you put a dye molecule at the one end and you put a quencher molecule at the other end. So dye is which would give out light. A quencher is which would absorb the light. So when this hairpin lobe would be in the hairpin form, no light will come out. So it's a self-reporting molecule. And if you introduce a perfect complementary sequence, this would open up and become double-stranded, which means the dye molecule has gone away from the quencher molecule. Light will start coming out. So we can play with these molecules and make them self kind of reporting, that if the target is there, if that bad gene that we're looking for, if it binds, it would open it up, it will result into lambda coming out. That's what I mean when I make lambda here. Right? So, and these quenchers and molecules are nanoparticles. Many of you have read on nanoparticles by now in KPR. So we bind nanoparticles to many of these. We use quantum dots with many of these. So quantum dot would be this dye molecule. Quantum dot would be the molecule that would give out light to the outside world, telling us if specific sequence is there or not, right? So these are some of the tricks. Questions?
Sir, what kind of information a DNA report has?
What kind of?
Information a DNA report contains.
So the most important is the presence of a gene in an unknown sample, right? So whether that's the DNA from the nucleus or whether we have taken the mRNA, which will tell us what is a cell making in itself. So we are now talking about an area called single cell analysis. Instead of looking at tissue where we miss, okay, our area, we have done lots of cancer work. So for us, or the oncologist, the most important thing is to find cancer in the early stages, which means to find cancer when one cell or few cells become cancerous, which means one or few cells start expressing proteins that they're not supposed to, or they start producing those proteins in excess amount than the normal expression, or they stop making something that would be required by that cell for example, for immunity, right? And hence the virus or the outside body can attack it or cannot attack it, depending on what's it. So that's where we want to see behavior of single cell or a few cells before we start looking at a tissue level or an organ level or a whole living thing, right? Human being or animals. So we want to look at molecular scale. And that's where nanotechnology comes in, right? Start getting some serious stuff, right? But I hope I can share this. Yeah. Go ahead.
Sir, for example, for cancer detection, the DNA sample,
Right.
what it would be? It could be any part of the body, or it would be a specific part.
Right. Okay. Now, many times when they do biopsy, when they find some cells, they take some of those cells out and they break apart and they get the genetic material out of those cells, which we are afraid of that they may be cancerous. What you're saying is very important, and that's what we feel that work needs to be done. That, think about it, we don't have a screening tool for cancer. Screening tool is like, I'll talk to you, Habib, just give me a second. Screening tool would be, we give urine sample, we give blood, and they find out if we have if we have high cholesterol, for example, right? So they have a test for cholesterol, but we don't have a test where we can look up if some cells or some molecules are running in our body, which may be indicating if there is cancer in some part. Or even there are cancer cells which start running in the bloodstream in the early stages of cancer, especially metastatic cancer. But they're too few, they get lost. So we are missing on that early detection kind of approaches there. Now, when you say, yes, you have to know the population or target population where we are looking for those sequences, right? Whole body is a big thing. We cannot, if we have a way of getting samples from different parts, saliva, urine, stool, blood, even the tears of from eye, they all carry lots of information that can be used to detect, if not DNA, but other enzymes and other uh, molecules that are where we can find out their overexpression or their downregulation, and that can tell us if things are good or bad. Right? Yeah, we'll go ahead.
Sir, I have two questions. Number one is,